Hello, everybody. My name is Sonny Hsiao. I'm the founder and CEO of Expodia. I graduated from National Taiwan University, major in chemistry, and then got my PhD from UC Berkeley. Today, it's my honor to reintroduce you our company, Expodia. Expodia is focusing on developing next generation off-the-shelf NK cell therapy. We focus on 4 Ps. First, our platform. We have 2 platforms. One is our off-the-shelf ONK cells. It's enhanced NK cells with desired marker profile for fighting tumors. And then we also have our proprietary ACC antibody cell conjugation technology, which allow NK cells to hit hard, hit fast, and hit often. We have a very strong team, and then we have the operations side, both in US and in Taiwan. We're very excited to develop a variety of different products that includes solid tumors and also liquid tumors, leukemia, and lymphoma. We also focusing on developing our partnership with pharmaceutical company and then also academia. So why the off-the-shelf NK cells will lead the revolution? There is why. First, NK cells have the strength over T cells, including no GVHD. NK cells have fewer inhibition mechanisms in comparison to T cells. And NK cells also have ADCC mechanism. They can synergize with antibody to fight tumor. And off-the-shelf NK cells also have other advantages, including quality, cost, and access in comparison to other cell therapy treatment. And then why Expodia we think we can be a very strong player in the field is because we have 2 platform technology. One is our ONK cells. ONK cells coming from our screening technology, NK Select. So our screening technology, NK Select, will allow us to generate most potent NK cells. And with the NK cell technology, we generate ONK cells have enriched activation receptors of NK cells, including CD16, NKG2D, and others. They also have minimized inhibition receptors, such as PD1 and TIGIT. In comparison to other companies, we also have another weapon, which is our ACC technology. As we mentioned, it can allow the NK cells to hit hard, hit fast, and hit often. And then it can also potentially overcome tumor microenvironment, which will be an advantage in solid tumor and in some indications of liquid tumor. The manufacture will be very simple and straightforward, that lower the cost and then be more affordable. We have both ACC and CAR technology so that we have 2 weapons for the NK cells and then also easier to make a dual targeted NK cells. So what's the key for the NK cell therapy to work? The answer is simple. You want the activated, active NK cells instead of exhausted or energy NK cells. So for that, the NK cells, for them to work, have the anti-tumor potency, they should have more activation receptors than the inhibition receptors. So how do you generate this desired NK cells become very crucial for the NK cell therapy. And this is what Expodia have developed, our NK Select technology, to generate the ONK cells with more potent, more activation receptors, and also minimize the inhibition receptors. So NK cell line donor, through our NK Select, we generate our proprietary ONK cells. ONK cells have many advantages over conventional NK cell lines, including the NK marker profile, stronger anti-tumor activity, minimal toxicity to normal cells, persisting of NK cell markers even after multiple cycles of freeze and thaw. So very stable. And then also it has been validated through standard mouse model. Here, if we're looking at the ONK cells in comparison to the conventional NK cell line, we can see that NK cells have enhanced activation receptors and minimal inhibition receptors, make it one of the best NK cells to fight cancer. On the other hand, we also have another weapon, which is called ACC. And then ACC can allow the NK cells to overcome suppressive microenvironment of tumor. It can hit hard, hit fast, and then hit often. So the concept of NK cell ACC is that we can use in ACC technology to arm the NK cells with more than 100,000 antibodies per cells. So our normal NK cells only have around 1,000 antibodies per cells. So NK cells have antibody attached to them. However, the amount is around 1,000. So in order to supercharge the NK cells, we developed the ACC technology that allows us to attach more than 100,000 of antibodies to the NK cells that make them to be supercharged and then very active to kill tumor cells. So that, as we said, it can make the NK cells to hit hard, hit fast, and then hit often. It's especially important for the solid tumor indications to combine our ACC to the NK cells. We're very excited to develop our pipeline so far. As we see, we have the pipelines targeting the solid tumor, and then we're positioned to be one of the first off-the-shelf NK cells against solid tumor. In addition to that, we also have our pipeline for B-cell malignancy, leukemia, and lymphoma. And then those are also going to the clinic very soon. And then we're very excited about those products. And then I will show you their progress. So first one, our ACE-1702, is an anti-HER2 antibody armed ONK cells. And this product, currently, we already initiated our clinical trial in 3 medical centers in the US. It will be a 2-stage of the clinical trial, phase 1, from dose confirmation to dose expansion. The treatment will be a lymphodepletion regimen followed by 1702 weekly dose for 3 weeks. For 1702, we're focusing on HER2 expressing tumor cells. Currently, there have been several drugs in the HER2 field. However, there are still huge unmet medical needs. For example, the women cancers, endometrial cancer, and ovarian. Currently, still there is no antibody or ADC approved for those indications. Same for the colorectal cancer as well. So those cancer type will be our main focus of our 1702. And actually, we have been shown 1702 have very good efficacy against those tumor types in the preclinical studies. So here is one example. We have generated the tumor mouse with the HER2 positive ovarian cancer tumor cells. And those ovarian cancer tumor mouse, we treated them with 1702. And then very exciting, we see that 1702 can actually shrink the ovarian tumor and then keep them at the baseline. We also did a comparison against current drug, which is antibody or ADC. And then for our preclinical study, we can see that against high expression HER2 tumor cells or low expression of tumor cells, 1702 has better efficacy in comparison to the current standard of care. As we say, using the ACC to make 1702 allow the NK cells to hit hard, hit fast, and then hit often. It armed the NK cells with more than 100,000 antibodies surrounding the NK cells. And then those antibodies act as a missile guiding system, guide the NK cells towards the tumor cell, activate the NK cell, and then kill the tumor cells, even under the suppressive tumor microenvironment, which includes low oxygen, high acidic environment, and then even in the ovarian cancer patient tumor ascites. Under those conditions, A1702 still can work very effectively. And then going to the next one, we also have our very exciting liquid tumor cancer drug as well, which is the 7055. 7055, we're using the CAR technology to put down the CD19 CAR on our ONK cells for the relapsed ALL and NHL patient. So this is a next generation CD19 CAR ONK cells with the CD28, CD3, and IL-15 signaling domain. And then the advantage of IL-15, especially for our ONK cell, is that it can activate the ONK cells, increase the potency, however, without the potential adverse effect, because our ONK cells will not be triggered to proliferate unlimitedly. It will be a very simple and straightforward manufacture process. And then we have validated the efficacy in the mouse model as of the MD Anderson. And here is the mouse data. We're using the same mouse model as the reference paper shown here. And then in comparison to cord blood derived CAR NK cells, which have shown human clinical activities, we're running the same experiment in the same mouse model, and then we see the very good activity of our 7055. So 7055 again is CD19 CAR ONK cells. When we apply them to the leukemia mouse, you see the efficacy even as early as day 5 of the treatment in comparison to the NK cell alone, and then also vehicle. If we're looking at the survival, it significantly increases survival. And if we run the comparison, it's actually slightly better than the cord blood derived NK cells. In the if regards to the increased fold of survival of the mice. And then we're also very happy to introduce our next product, which is 1975. It's a product that target CD70 positive liquid and solid tumor. 1975 will be a CD70 antibody armed ONK cells for AML and then also RCC. In this case, ACC is chosen over CAR because ACC again is supercharged, hit fast, hit hard, and then hit often. So it can potentially eliminate leukemic stem cells. However, not wipe out all of them, which will be cause severe side effect of myelodepletion. So this is our third product in the pipeline. The CD70 marker that can target and kill leukemic stem cells and then hopefully to treat AML patient.